We now have Yash Agarwal from the Massachusetts Institute of Technology in Genti Bio, and he will be presenting on a materialist-based approach for localized delivery of cancer immunotherapy.
Great. Thanks for the introduction, Isabel. Can you see the slide okay?
Yes, we can see the slide.
Perfect. So yeah, thanks for inviting me for this talk and this opportunity. My name is Yash, and today I'll be presenting some of my PhD work that I completed at MIT in the labs of Dane Wittrup and Daryl Irvin. And the topic of the talk today is enhancing local delivery of cancer immunotherapy with the vaccine adjuvant alum. Before I begin, I wanted to just touch on the different treatment modalities that have existed for cancer historically. Oftentimes, cancer will manifest as these tumors that take over healthy organ function. From the very beginning, the most obvious solution has been to just gut the tumor out by surgery. And while this has been highly effective in reducing tumor burden, even microscopic disease left behind can come back and become more dangerous. Going forward, people started to hit tumors with X-ray radiation to kill the cancer cells directly. And now standard of care has evolved to intravenous treatments with chemotherapies. While both have shown to preferentially kill cancer cells, there is still quite a bit of damage to healthy cells, which can lead to a lot of unpleasant side effects like pain, fatigue, GI problems, and more. And something that none of these therapies exist or address is that cancer is partially a disease of the immune system. Here I'm showing some hallmarks of cancer, and you'll notice that avoiding destruction from the immune system is a very important step in cancer progression. Somehow cancers have evolved to evade recognition and attack from key immune players like T cells, NK cells, and dendritic cells. What we often see with tumors is that either T cells can't get into the tumor, or once they do, they're essentially inactivated or exhausted. A new class of cancer treatments that has recently gained a lot of prominence in the clinic is cancer immunotherapy. And cancer immunotherapies can reprogram our immune system to recognize the tumor as foreign and attack only tumor cells without killing healthy cells as much. Specifically, checkpoint blockade antibodies like anti-PD-1 can prevent T cell exhaustion in the tumor, keeping them active for longer. And these have shown to work really well in a variety of different cancers in clinical trials. Here I'm showing some data from a non-small cell lung cancer trial where on the y-axis you have percent people remaining in the study whose tumors haven't grown out. So if a therapy cures 100% of the people, you'd see a straight horizontal line at 100 over here. You see that over time, checkpoint blockade therapy can significantly improve patient outcomes over chemotherapy. Even so, checkpoint blockade only seems to impact or improve outcomes in a subset of all patients, not everybody. One hypothesis is that these therapies don't work as well if there aren't enough T cells in the tumor to begin with. So one approach to improve efficacy is to combine these with other immunotherapies. Combinations, particularly with inflammatory cytokines like IL-2 and IL-12, can improve T cell infiltration and activation. And together, these therapies have been shown to eliminate tumors that were previously non-responsive to checkpoint blockade alone. For my work, I'd like to focus on IL-12 a bit. IL-12 is a heterodimetic protein, and as a cytokine, it has been known to activate various steps in the cancer immunity cycle. In particular, it can directly activate T cells into becoming better killer cells for cancer cells, while also indirectly promoting more tumor antigen-specific T cell infiltration into the tumor. So clinical trials where IL-12 was given directly in the blood or IV first started in the 1990s. However, early results for systemic IL-12 were disappointing. Too little of the protein got into the tumor where we wanted it to activate T cells, and too much of it went everywhere else. So you had side effects like flu-like symptoms, severe gastrointestinal distress, and liver toxicity as only some of the few symptoms that people had. So key question where this project began was how can we improve the delivery of IL-12 to only tumors and not healthy organs to kind of localize delivery of this cytokine. One idea is to directly inject the protein into the tumor using intratumoral administrations. Recent advances in interventional radiology, endoscopy, and laparoscopic procedures have greatly our ability to access essentially any lesion within the human body. And so intratumoral therapy has become a viable option for the dosing of many types of drugs. However, while the expectation is that everything we put in a tumor stays in there, in reality, a lot of it will leak out. So to prevent that leakage, we look toward biomaterials for solutions. The material we settled on is alum. Alum, or the aluminum hydroxide adjuvant, is an FDA-approved vaccine adjuvant and is already administered annually to millions of people in over 20 clinically licensed vaccine formulations. The material at its base exists as small nanorods that come together to make larger micron-sized aggregates. And this size becomes really important as we start thinking about particle trafficking in vivo. Any particles that are smaller than about 100 nanometers are very susceptible to leakage through the lymph or blood. And even the biggest proteins are much below that threshold. So we hypothesize that if we can engineer strong protein binding to alum, we can use the alum protein complex to dramatically limit leakage of the therapy or drug through the tumor blood vessels after intratumoral injection. So how do we do that? How do we improve protein binding to alum? When we looked in the vaccine literature, we found that phosphorylated proteins with phosphotags, bound to alum much better in vivo. So we engineered that. We engineered a phosphorylated tag on IL-12. We call that the alum binding peptide. And here you can see that in vitro, the phosphorylated version of IL-12 in purple binds to alums at high amounts, while unphosphorylated variants don't. Then we want to track whether this also happens in vivo. So we fluorescently labeled IL-12 and then directly injected that into tumors with or without alum. You'll see here that when we track in vivo protein retention by IVIS imaging, we see that alum-anchored IL-12 in purple remains at the tumor for a few weeks after treatment, while unanchored IL-12 shuttled out within a few days. So as this IL-12 leaks out, is it promoting toxicity systemically? From the literature, we knew that systemic IL-12 toxicity can manifest with various symptoms in people and mice. Two that I wanted to highlight is liver dysfunction. So liver toxicity caused by systemic IL-12 can lead to a higher concentration of some liver enzymes, particularly ALT in the blood. And IL-12 can lead to severe weight loss and emaciation in mice and people. And it's exactly what we see. Intratumorally injected IL-12 alone leaks out, and it can promote significant weight loss in mice while also leading to liver toxicity. You see ALT levels go beyond the clinically healthy levels and get really high. While when you mix the same protein with alum and then inject that into the tumor, you eliminate the weight loss while bringing the liver tox or ALT levels back into clinically manageable levels. So our engineered cytokine alum combination can dramatically diminish IL-12 related systemic toxicities seen after an intratumoral local injection. Next, we wanted to analyze how alum-anchored IL-12 could potentiate the efficacy of existing therapies like anti-PD-1 in a model that doesn't typically respond to anti-PD-1 alone. So we picked the B16F10 melanoma model. It's a pretty common model that cancer immunologists or people working in cancer translation use. We treated established tumors with a single dose of IL-12 followed by systemic anti-PD-1. While IL-12 alone promoted little tumor growth delay, so you do see a little bit of effect with just IL-12, the alum-IL-12 combination led to dramatically better responses. Tumor growth delay was increased by weeks, while we saw complete responses in the majority of the mice with this really hard to cure model. Since we had these responses, we then wanted to challenge our therapy further in a new model. This is where I got to collaborate with another grad student in the lab, Anthony Tabbitt, and we wanted to see if we could treat brain tumors directly with our intratumoral alum treatment. So we inoculated glioma cells directly in the brain, and a few days later treated those tumors with either cytokines alone or cytokines mixed with alum. Again, we see very similar results to what we observed in the flank. Unanchored cytokines led to dramatic toxicity in these mice, so much so that we had to euthanize about 50% of them early in the study, while alum binding eliminated that phenotype while maintaining tumor control in purple. So in another model, alum-bound cytokines were less toxic, more effective. And in the same brain tumor model, in another experiment, we can also observe the behavior of mice with tumors using an open field test. Essentially on day 3, you notice that mice in both cohorts, the negative control or the mice that got alum cytokines, are very active, exploring their surroundings, indicating strong locomotor activity. However, just a few days after, mice that got saline become very stationary. And this video is on, they're just not doing anything. While the mice that got alum and cytokines, they're still excitedly exploring their environment. This makes sense when we look at the brains of these mice. The tumor inhibits regular brain function in the mice that got a negative control or vehicle treatment, while the alum cytokine treated mice had barely any tumors, and they can still explore their surroundings pretty well. And we can also quantify this with a few mice, and it becomes obvious that the tumor clearly impacted average velocity and total distance traveled, and that our therapy could help dramatically reduce that. All right. Hopefully, I've shown you today that by incorporating phosphorylated tags on therapeutic proteins, we can improve their binding to alum in vivo, such that even a single dose intratumoral treatment can have dramatically improved preclinical results in a variety of models. If you liked what I've shown you today and want to read more about our work, many other models that we worked on, the mechanism of action investigation that we did, this study was published in Nature Biomedical Engineering last year. So with that, I'd like to acknowledge both my PIs and labs for help during the PhD, some funding sources, and also thank the organizers for inviting me to present my work. Thank you. We can take any questions now.
Thank you so much for the talk, Yash. Now I believe Shivali has a question.
Hey, Yash, nice
Hello.
to see you. Yeah, great talk. This is really exciting, and I really liked how, as I had questions, honestly, you answered them in your talk. Very well structured. I do want to ask, so you were able to sort of supercharge these tumor infiltrating lymphocytes and get that inflammatory phenotype back on. Did you look at persistence of how long that phenotype can remain locked in that tumor microenvironment? And, you know, is that something that needs to be addressed with multiple dosing cycles, or something that changes with body weight as you think about scaling into humans, things like that?
Great question. And that's something we think about a lot in the mouse, in particularly the model that we work with. We see strong interferon gamma responses in the tumor induced by alum-bound IL-12 for as long as 7 to 8 days after treatment. But they do eventually diminish. I think at some point, even though we see the protein with IVUS imaging for weeks, they're not really active. I think what we really need is that first week of strong activation to boost that efficacy. But I think as we translate this into the clinic, we are thinking a lot about, okay, do we want to have cleavable linkers in there where we could actually cut out the IL-12 after some time? Do we want to redose if the IL-12 becomes inaccessible? So I think that'll be a very model-to-model, patient-to-patient specific design.
Gotcha. Thank you so much. And I think we have a couple other questions as well. I'll
Slide away.
Hi, Yash. Great to see you. Long time no see. Really, really nice talk, even though I heard some of this work a little bit before. I was curious, actually, one thing about the B16 model was quite aggressive.
Yeah. And
you had about 50% survival with the alum IL-12. Do you think it actually is undermining a little bit the potential efficacy of your drug and delivery system because of just the way the model is set up and how quickly it can become very aggressive?
I mean, yeah, so that's part of why we tested our therapy in various other models. So when we look at a colon carcinoma model like MC38, we get close to 100% cures. So I think, as you know, with any preclinical models, it all depends on what you set up. And to show any therapeutic efficacy, you do want to show that in many different types of models. That's why we have brain cancer data, colon cancer data, skin cancer data. We also have data where we put 2 tumors in a mouse and treated 1 tumor, and we see strong efficacy, even with our highly localized therapy tumor. So yeah.
And I know this lab was really the focus is cancer, but do you know if this has been tried at all in the context of autoimmunity, autoinflammation?
No, it's come up a bunch, especially because we have a platform and you can put whatever you want on it. So you could put cytokines of therapies that would help with maybe Treg expansion, et cetera, in the context of autoimmune disease. I personally don't know if the lab's now working on it or not, but I do know that this has spun out into a company, and that's something that they might be looking into.
It's fascinating. Thank you.
Thanks.
I have just one quick question, if I may.
Of
So beyond, limiting the egress or containing IL-12 in the tumor microenvironment, alum, as you rightly pointed out, is used as an adjuvant.
Yep.
Toxin. So what is its role beyond just egress? I mean, presumably you looked at that.
I spent a year looking at that, at least in our models.
Yeah.
Huh?
Is it activating the immune system?
Yeah, so I think what it's doing is that it is definitely keeping the IL-12 in the tumor for longer. We really dug into the inflammasome pathway. We dug into various other, like the IL-1 secretion pathway, et cetera, that alum has been known to take part in. And none of those effector molecules appear to play a role. So when we do this therapy in NLRP3 knockout mice, they work just as well. When we do this therapy in mice that were had neutralizing antibodies given for IL-1 beta or IL-18, no real change. So it does seem to, at least in the models that we looked at, alum had a purely retention aspect. But I think long term, we also discussed a lot how much alum should we give. Maybe it's a dose-dependent effect. If we give more alum, maybe that's when we start seeing more of that happen.
Thank you.
Thank you.